Hello, my lovely anatomists and physiologists. Michele here, and we are finishing up our chapter 19.2 conversation here, looking first at cardiac muscle, and then we'll be moving on to what's described as the pacemaker or pace setter system. We talked about cardiac muscle. Let's remind ourselves of the cell. So it's going to be sort of a small cell in compared to like skeletal muscle. And we're going to see that this cell is described as branched. So I'm drawing two vertical lines and then sort of like a V shape, kind of like in between on each end to represent the branching. And then I'm going to do a single nucleus in the center. And then I'm going to do kind of like my zipper Z lines on the ends of the cell. And then I'm going to include my striation. So we will see that cardiac muscle cell does have a striation. It is striated. So that's going to mean that the actin and myosin is organized in the cardiac muscle cell in the same way that we talked about for skeletal muscle. And what we'll see about these cells is that they do have the single nucleus. They do have a lot of mitochondria, which is of course our location for aerobic respiration, meaning using oxygen to break down glucose or fatty acids in order to make that ATP molecule. Now, when I drew sort of my zipper kind of lines at the ends of my cells, what I was representing there is my intercalated disc. And the intercalated discs are features that are unique to our cardiac muscle. So whenever I teach how to recognize this tissue in the lab, I always say, look for the intercalated discs. Now, the significance of the intercalated disc is that it's going to join connecting cells. And it's going to be a join that includes what are called desmosomes. And you can think of these as little like physical welds in between these cardiac muscle cells. And then we also see what are called tight junctions. And tight junctions are going to be little like watertight seals between cells. And then we're going to see what is called the gap junction. So when we talked about the desmosome and the tight junction, this is a physical connection between the cells. When we talk about a gap junction, this allows for the movement of ions, which remember means signal, movement of ions between cells. And so collectively, this intercalated disc with the desmosomes and the tight junctions and the gap junctions provide what we can describe as a functional syncytium. Functional syncytium. And what this is saying is that rather than cells functioning independently of each other, in the case of the cardiac muscle tissue, we can consider that the cells basically are collectively working as one. So all of the cells of the top half of the heart, the left and right atria, are functioning kind of like one massive cell. Essentially, they can go 1, 2, 3, go. And the signaling contracts together, the connections between pull on each other, so they're functioning like one. So this makes a very effective pump. We can have the same sort of idea for the lower half of the heart. So the left and right ventricles can then basically go like 1, 2, 3, go, and all contract together. And that's because of these intercalated discs. This is a very important feature. Okay, so we've already mentioned that we have lots of mitochondria. So we are doing aerobic respiration, which remember does mean that we're using oxygen. When we talk about the type of contraction that we get a twitch type of contraction. And we describe it as having a long refractory period And then we'll also say it has a short relaxation period. And when I talk about this feature of the cardiac muscle tissue, I like to describe it as like a contract and hold, or like a squeeze and hold. So think of taking a wet sponge and really like doing one squeeze and like holding it for a period of time to really wring out, to really get as much water out of that sponge as you can. So the heart, these cells are contracting, producing this strong squeeze and hold to really effectively get that blood pumped out through. This long refractory period is going to be important in actually preventing tetany, which is something that your skeletal muscle can do, but your cardiac muscle can't. And that's good, because if we had tetanic contractions in the heart, the muscle would contract and stay contracted rather than like a squeeze and hold and another squeeze and hold and another squeeze and hold. So we don't want it to stay contracted. The other thing that we see with the long refractory period is that we're going to use calcium ion channels to help give us what's going to be described as a long plateau, meaning when you graph out the muscle, cardiac muscle contraction, you'll see like that long refractory period. Another important feature of the cardiac muscle tissue is that it has what's called autorhythmicity. Autorhythmicity. And so this is where it can initiate its own electrical signal. We will see that the cardiac muscle is going to be controlled by both the autonomic nervous system and the endocrine system. But even in the absence of these two body systems, the cardiac muscle tissue will create its own signal. So we're going to have cells that are going to do what's called spontaneous depolarization. And this spontaneous feature means it's not depolarizing because of a signal from the autonomic nervous system or the endocrine system. It's doing it on its own. And it's doing it on its own because we'll see that you have special cardiac muscle cells that have sodium ion leaving the cell, so that it's going to go from a -60 millivolts spontaneously to a -40 millivolts. And then what we'll see happening is at -40 millivolts, you get calcium ion channels that will open. And then this is going to give you your depolarization for your action potential, which, remember, is our name for like the signal, right? When we talk about the long refractory period and then the short relaxation, we'll see that it's going to be potassium ions that are leaving the cell in order to repolarize the cell. So it's a movement of sodium, a movement of calcium ions that depolarize the cell. So let's write this down. So when we talk about a cell being depolarized, that is stimulated. That's happening from the movement of sodium ions and or calcium ions into the cell. And when we talk about the cell kind of returning to rest, we call that repolarizing. And that's going to be the movement of potassium ions out of the cell. So that's just kind of connecting information that we've learned in Anatomy and Physiology Part 2. when we talk about cells that are having this autorhythmicity, when we talk about these cells that are spontaneously depolarizing, what we see is that there's actually two types of cardiac muscle cells. So these cells that are doing this are called myocardial conducting cells. So myocardial conducting cells are cardiac muscle cells that have autorhythmicity, that can spontaneously depolarize. When we're talking about the cardiac muscle cells that are really pumping the blood, so let's talk about them down here in terms of the twitch type of contraction with the long refractory period and all that. Let's talk about these as myocardial contracting cells. And so these are the ones that are really like pumping the blood. Okay. All right, up next, let's look at the relationship of the myocardial conducting cells together and what is described as the pacemaker or pace setter system. As always, take care of yourselves and each other.